Hi everyone. So today we'll talk about citric acid cycle, or which is also known as TCA cycle or tricarboxylic acid cycle, or it is also known as citric acid cycle or Krebs cycle. Krebs is because it was discovered by Hans Krebs, so the name is Krebs cycle. So to give you a overview of the cycle, it is a pathway for generation of energy equivalents, reducing equivalents in the form of ATP, and this is a type of oxidation reactions where all the oxidation reactions are the energy is stored in the form of reducing equivalents like NAD plus or NADH2, FADH2, etc. So what happens is glucose is converted into pyruvate and then acetyl coenzyme A in glycolysis. So in other metabolic pathways, fatty acid is converted into acetyl coenzyme A. Amino acids, which are also one of the major macromolecules, biomolecules which are present within the cell. So these amino acids and fatty acids, they are converted into acetyl. Fatty acid is converted specifically into acetyl coenzyme A. Glucose is converted specifically into acetyl coenzyme A. First, glucose is converted into pyruvate, and then from pyruvate, the acetyl coenzyme conversion of pyruvate to acetyl coenzyme A is carried out by the enzyme Pyruvate Dehydrogenase. And amino acids, it can be converted either into pyruvate or acetyl coenzyme A. If it is converted directly into acetyl coenzyme A, then it enters the TCA cycle. However, if it is converted into pyruvate, then again by the enzyme Pyruvate Dehydrogenase, it is converted into acetyl coenzyme A, which then enters the TCA cycle or tricarboxylic acid cycle and generates reducing equivalents, carbon dioxide and the electrons. Now these electrons then undergo electron transport chain and oxidative phosphorylation for the conversion of ADP, adenosine diphosphate, into the energy currency, which is ATP, adenosine triphosphate. So this is the general overview of TCA cycle and some of the alternative names. Then again, this cycle is amphibolic. And what is amphibolic means? It is both catabolic as well as anabolic, because some of the intermediates which are generated in the cycle, which we'll discuss later on. So some of the intermediates of the cycle, they are used in some other metabolic pathways in the cell. Also, this cycle requires aerobic conditions. Okay, so this cycle is strictly aerobic. Under anaerobic conditions, what happens? The energy production, the cell does not produce sufficient energy, so more energy is generated under aerobic conditions than in anaerobic conditions. And what is aerobic means? It requires oxygen. Anaerobic in the absence of oxygen, aerobic in the presence of oxygen. Okay, so if the cell is under anaerobic energy production mode, then production is not that sufficient, and only 10% of the pyruvate generates energy. So there is very less amount of ATP which is produced under anaerobic conditions in the cell. Okay, so some of the coenzymes that are reduced during this TCA cycle are NAD plus or to NADH, FAD into FADH2, and these reduced coenzymes then they are used by the electron transport chain and oxidative phosphorylation for generation of ATP. Okay, now this TCA cycle serves two purposes. There are two purposes of this TCA cycle which it carries. So first, generation of reduced coenzymes. These reduced coenzymes, they are used to produce energy. So acetyl coenzyme A, which is the starting material for the TCA cycle, is used for generation of ATP, CO2 and FAD into FADH2 and NAD plus into NADH. Okay, and then the second is it supplies various intermediates for the biosynthesis of carbohydrates, lipids, amino acids, nucleotides, etc., which are other major biomolecules which are present. So the intermediates, they are used, all the intermediates which are generated in the cycle, they are not wasted, they are used in some or the other metabolic pathways. Okay, so, and this cycle, there are, at every step, there are certain modulators. Modulators are of two types. Either they repress or they enhance. So there are two types of modulators. The presence of some of the modulators, they may slow down the rate of the reaction, or they may slow down the cyclic process in forward direction. So they are known as repressors, and the presence of those modulators, they increase the speed of the cycle. So they are known as activators. So the modulators again are of two types, repressors and activators. Alright. So now this is the detailed, complete pathway of the citric acid cycle, which is composed of 8 steps or 8 reactions. So it starts with pyruvate. So first, pyruvate is converted into acetyl coenzyme A in the presence of the enzyme pyruvate dehydrogenase. Okay, and at this step, so here the plus sign, plus and minus, so the positive ones, these indicate the activators of those so the presence of these molecules within the cell cytosol will increase this reaction, or it will increase the conversion of increase the speed of conversion of pyruvate to acetyl coenzyme A, while these minus, so they are repressors. So the presence of these will slow down the speed of this reaction. Okay, so if we look into the first step, so here pyruvate is converted into acetyl coenzyme A in the presence of the enzyme pyruvate dehydrogenase, and the activators of this step are AMP, adenosine monophosphate, coenzyme A, and NAD+. However, the repressors are ATP, acetyl coenzyme A, and NADH. Means if these ATP or acetyl coenzyme A, they are present in excess number than pyruvate, then first the cell need to consume the present acetyl coenzyme A through this reaction, through the cycle, then only the rest of the pyruvate will be converted into acetyl coenzyme A. That is why the presence of these molecules will act as repressors, and these will decrease the rate of conversion. Now this acetyl coenzyme A then enters the citric acid cycle, and it is the first step, it is converted into citrate. The conversion of acetyl coenzyme A into citrate is catalyzed by enzyme citrate synthase, and here ADP, adenosine diphosphate. Remember, this ADP acts as activator. However, the repressors are NADH, succinyl coenzyme A, ATP, and citrate. Now this citrate is then converted into isocitrate, and then this is an isomerization reaction, and then which is carried out, sorry, I forgot to write down the enzyme here. So it is carried out by isomerase. Then this isocitrate is converted into alpha ketoglutarate, and the conversion is catalyzed by enzyme isocitrate dehydrogenase, and here again ADP acts as activator. However, ATP, adenosine triphosphate, which is the energy currency of the cell, acts as repressor. Now this alpha ketoglutarate is then converted into succinyl coenzyme A, catalyzed by enzyme alpha ketoglutarate dehydrogenase, and succinyl coenzyme A and NADH, these act as repressors. Now this succinyl coenzyme A is converted into malate, and during this conversion FADH2 and GTP, which is then converted into ATP, they are generated, and the conversion of succinyl coenzyme A to malate is catalyzed by enzyme succinate dehydrogenase. Now this malate is again then converted into oxaloacetate, catalyzed by enzyme malate dehydrogenase, and then this oxaloacetate then again enters the next round of the cycle. So this is all about citric acid cycle, and all the enzymes, all the steps involved, all the enzymes and the activators and repressors. Now, so there are some key differences between the citric acid cycle and glycolysis. So glycolysis is the first and foremost thing is, remember glycolysis, it is a linear pathway. It is composed of 10 steps. And whereas this citric acid cycle, this is a cyclic process. So oxaloacetate again then enters the next round and with acetyl coenzyme A forms citrate. So it is a cyclic process. Then glycolysis occurs in the cytosol, and this TCA cycle, where it occurs in the mitochondrial matrix. Then glycolysis does not require oxygen. It may be aerobic or anaerobic. However, this is strictly aerobic. This cycle is strictly aerobic. It means it requires oxygen. Why? Because oxygen acts as the terminal electron acceptor and is converted into water. Remember the first cycle which we draw in at the beginning of the lecture. Okay, so to summarize this cycle, so if we summarize this entire cycle, what we get is that for each acetyl coenzyme A, for one acetyl coenzyme A which enters the citric acid cycle, 2 molecules of carbon dioxide are generated. So we have 2 CO2 which is released. Then we have coenzymes NAD plus and FAD that are reduced. These coenzymes are reduced. Then we have 1 GDP or ATP, which is phosphorylated into GTP or ATP. And then fourth is that the initial acceptor molecule oxaloacetate, it is reformed, and then again it enters the next round of the cycle. So this is the summary of the citric acid cycle. Okay, so now how the energy is generated in this TCA cycle. So energy is generated here in the form of energy stored in the form of reduced coenzymes, and then they are further converted into ATP by these reduced coenzymes are consumed in the electron transport chain and oxidative phosphorylation, and then they are converted into ATP molecules. So here in the TCA cycle, the energy is stored in the form of reduced equivalents. Now for each acetyl coenzyme A, for one acetyl coenzyme A which enters the TCA cycle, we have 3 NADH. We have 3 NADH which is generated. Then we have 1 FADH2 which is produced, and we have 1 GTP or ATP. So for 1 NADH, we have 2.5 ATP which is produced. For oxidation of 1 FADH2 generates 1.5 ATP molecules. So overall, how many ATPs will be produced? So we have 3 NADH. 1 gives 2.5 ATP molecules. So the complete oxidation of 1 acetyl coenzyme A will give us 10. So total 10 ATP molecules will be produced by conversion for 1 acetyl coenzyme A which is entering the citric acid cycle. Total 10 ATP molecules are produced. So if we summarize the aerobic oxidation of glucose, first by glycolysis, then TCA cycle, and then so how many ATP molecules will be produced? So during glycolysis, how many ATPs? So if glucose is converted into 2 molecules of pyruvate during glycolysis, we have 2 NADH which is produced, and here we have 2 ATP which is generated. Then this pyruvate is converted into 2 acetyl coenzyme A. Now 1 generates how many? 10. So 2 will generate 20 ATP. How? 20. So 2 acetyl coenzyme A will enter the citric acid cycle or the TCA cycle, and it will generate 6 NADH 2 FADH2, and here we have 2 ATP or GTP. So 2 plus 2, here यहाँ पे हमारे पास we have 4 ATPs, and here 2 NADH. 1 NADH gives 2.5. So total we'll get over here 28 ATPs here. So total we'll get 32 ATPs by complete aerobic oxidation of one glucose molecule. Complete aerobic, remember this term, aerobic. Complete aerobic oxidation of glucose will generate 32 ATP molecules. Whereas anaerobic is in the absence of oxygen. So anaerobic, lactate is formed from pyruvate, not acetyl coenzyme. Lactate is formed from pyruvate after glycolysis by lactate dehydrogenase, and one NADH is formed. So total by anaerobic oxidation, you only get net gain of 2 ATP per glucose as compared to 32 in case of aerobic one. So anaerobic is only 5 to 10% efficient in terms of ATP production as compared to the aerobic. Okay, so but however this anaerobic respiration occurs, or anaerobic degradation, it occurs when there is extra demand. There is so much demand of ATP that the cell cannot produce them by aerobic oxidation. So then to meet the high energy demand, so the cell undergoes anaerobic oxidation or anaerobic degradation. Like it occurs in muscles during exercise because they go into oxygen deficiency. Then metabolism in muscle returns to normal when oxygen is replenished. So this is all about citric acid cycle, and if you all have any queries or doubts, you can connect with me on my email ID which is there, or you can also post in the comment box. I'll be happy to answer all your queries and doubts. Please subscribe to the channel if you have not yet subscribed for more such informative videos. Do hit like and share it among your friends. Thank you.